Hello, grade 9. Hope you're doing well. Today, we're going to continue Chapter 8, Conformed Reproduction of Genetic Information, with Activity 2, Conformed Reproduction of Chromosomes. So please open your books on page 160. So let's start with a quick review of mitosis. What is mitosis? So mitosis is the process by which your body cells divide more and more while conserving the same number of chromosomes and the same genetic information. But you know that, first of all, every cell starts a phase of interphase or growth phase, during which chromosomes become duplicated. Here, the chromosomes are still in the state of chromatin, long and thin. The centrosome is still visible in the cytoplasm, the nuclear membrane always present, and as you see, I can't see the chromosomes very well, I can't count them. Next, the cell starts mitosis. In two phases, the first phase of mitosis, the chromosomes that are now duplicated with 2 chromatids each condense, that is, they become thick and visible, and we can count them. The nuclear membrane starts disappearing. The asters now are present. Where do they come from? Yes, it's the centrosome that divides into 2 asters that join together with the spindle fibers. What happens in the next phase, metaphase? These chromosomes are now in the cytoplasm, so the nuclear membrane has disappeared. They join, they gather in the center of the cell, forming the equatorial plate. And, of course, every chromosome fixes to every spindle fiber by the centromere. Anaphase, the third phase of mitosis, every chromosome of those found in the center of the cell divides into 2 chromosomes of 1 chromatid each. And this is what we call here polar ascension, characterized by migration of the chromosomes toward the opposite poles of the cell. Mitosis ends by telophase, where this cell constricts, divides into 2 daughter cells, each with 2 N chromosomes, 1, 2, 3, 4, that are 2 pairs of chromosomes, but of course every chromosome with 1 chromatid. What happens to these 2 daughter cells now? Yes, again, every daughter cell of these undergoes a new interphase, a new growth phase, so it can again divide more and more until we get a large number of cells. Okay, so what are the objectives of this document? The first objective is to compare mitosis in animal cells and in plant cells. Second, it is to compare the karyotype of the zygote with the karyotype of 4 and 8 cell stage embryo. Third, to conclude that all body cells except gametes have the same karyotype. And fourth, to explain the importance of the duplication of genetic material of chromosomes before every mitosis. So, let's do a comparison between mitosis in animal cells and mitosis in plant cells. So, as we know, in animal cells, animal cells have what we call a centrosome. And this centrosome divides into 2 asters, and these asters have an important role during mitosis. So, first of all, animal cells are characterized by the presence of asters. However, plant cells are cells with no centrosome. So, consequently, there won't be any aster in mitosis. So, no esters during mitosis in plant cells due to the absence of centrosome. We move to the second difference. We know that telophase happens in animal cells by a cleavage furrow that takes place, which is a constriction of the cell membrane from outside to inside that leads to cytokinesis. Cytokinesis, which means a division of the cytoplasm of the mother cell into two daughter cells. However, if you look at telophase in plant cells, you see clearly that no constriction, no cleavage furrow is observed. However, there is a cell plate that forms in the center of the cell, and this cell plate grows more and more until it divides the cytoplasm into two cells, and then this cell plate forms the cell wall that doubles the cell membrane. So the second difference is a cleavage furrow during telophase of the animal cell, while no cleavage furrow during the telophase of the plant cell. However, there is formation of the cell plate in the middle of the cell. Okay, we can start now with the document 2 or activity 2. You know, the zygote is the cell or the egg cell that results from the union of the male gamete, the sperm cell, and the female gamete, the ovum. So the zygote has 2 N chromosomes or 23 pairs of chromosomes or 46 chromosomes. By successive divisions and successive mitosis, of course, the zygote was able to produce all body cells and consequently all body organs that you see in this figure: the lungs, the liver, the intestines, the bladder, kidneys, and all these organs, of course, form the human being. You see that every organ of these organs, which is made up, of course, of cells, present 2N chromosomes, which is the same number of chromosomes as the zygote. And this is the characteristic of mitosis, which is the conservation of the number of chromosomes. We're going to answer these 2 questions. Do the cells of the body carry the same karyotype? And how are chromosomes reproduced? Okay, so please, to answer these questions, I want you to open your books on page 160. Here, the 3 figures that you see documents ABC. The first one corresponds to the zygote and its karyotype. The second one to a 4-cell stage embryo, an embryo made up of 4 cells, and its karyotype. And then in document C, this is an embryo made up of 8 cells, and this is the karyotype of each cell of this embryo. Okay, my question here is compare the karyotypes. Of course, how would you answer this question? To compare these karyotypes, you should look and observe very well these 3 karyotypes and find the similarities and the differences. Okay? So, do you see any differences? I think no differences are observed here between these 3 karyotypes. If you count them, the 3 karyotypes show 46 chromosomes or 23 pairs, and these chromosomes are identical in the 3 karyotypes I mean that pair 1 in the first karyotype is the same as pair 1 in the second karyotype, the same as pair 1 in the third karyotype, and so on. Moreover, the last pair, which is the gonosome, is XX in the first, XX in the second, XX in the third. So, by comparing these three karyotypes, no differences are observed. The three karyotypes show the same number of chromosomes as well as the same structure. What can you conclude? We conclude that, okay, all body cells possess the same karyotype. Of course, when I say all body cells, we don't mean the gametes or the sex cells. Even if I take a cell from the intestine, a cell from the lungs, a cell from the muscles, any cell you're going to choose and you're going to do its karyotype, you are going to find that the karyotype is always the same, 23 pairs, 46 chromosomes, all autosomes are identical to each other, and all karyotypes carrying the same gonosomes, of course, if these cells are taken from the same individual, from a woman or from a man. Let us answer the second question. How are chromosomes reproduced? The confirmed or identical reproduction of chromosomes takes place during interphase. How? Look at the figure below, please. At the beginning of interphase, each chromosome that is long and thin has one chromatid, as you see here in the figure. Duplication of chromatids occurs and condensation starts. Thus, we obtain a duplicated chromosome that condenses more and more. The duplicated chromosome now, as you see here, is of two chromatids that are attached together by the centromere, and now this duplicated chromosome is ready for division. This is document E from your book on page 161, which explains very clearly how chromosomes reproduce in a conformed way. What do we mean with conformed reproduction? It means that every chromosome makes an identical copy of it. Look at the document, please. This is the duplicated chromosome at the end of the interphase. It condenses, and then during mitosis, and mainly during anaphase, as you can see, it divides by splitting of the centromere. So we obtain two chromosomes, each of one chromatid. Then each chromosome of one chromatid, found now in every daughter cell, of course, will, as you can see here, during interphase, duplicate again, and so on. This duplicated chromosome again will start a new division, a new mitosis. Okay, so to conclude, by successive divisions, the zygote produces all the cells of the body. All these cells, of course, always remember, except gametes, have the same karyotype. During interphase that separates two successive mitotic divisions, each cell duplicates its chromosomes. Hence, the genetic information of the cell is also duplicated during interphase. And it is this duplication that allows the conformed reproduction of chromosomes. Please do the following homework, exercise 1, and also exercise 2. Please study very well the lesson and do the homework. See you later. Thank you for your attention.